thank you very much for the very kind introduction, and thank you, Annika, and the team really at CPM and OxCODE for organising this event. It's really lovely to see so many people here. Can I just ask a quick show of hands? So who of you are medically healthcare qualified, you know? Okay, and who of you all are interested patients, participants, you know, sort of advocates today? That's about 50-50. That's fantastic. Okay, so will you put your hand up when I start saying something you do not understand? Can you be the... Okay, and Annika as well. Okay, so I'm just going to be talking about the work that we've been doing and trying to sort of introduce the session today. So we're trying to develop cancer prevention vaccines. And we literally mean that. We want to give vaccines to people who do not have cancer to protect them from getting it in the future. So much in the same way that we vaccinate children now, you know, against different infectious diseases, we want to be doing exactly the same thing in the future and do it at population level, protect people from cancer. Because I really don't understand why we need to get cancer. The incidence is rising, and this is becoming a crisis. They talk about this being the silver tsunami. Because we are aging, we are living longer, and we are living more unhealthy lifestyles. And so it's estimated that by 2070, there will be 34 million cancer cases worldwide, which is about the population of Canada getting cancer every year, which is quite inconceivably large. And we know with the way that we have, you know, from the oncology perspective, the earlier you get in to treating cancer, the better the outcome. So this hasn't projected all the little people very well. But in the example of lung and ovarian cancer, two typical cancers that actually, if you diagnose them at early stage 1, where they're just confined to the lung or just to the ovary, your chances of cure are so significantly greater. And the shocking thing that I find that still surprises me is that cancer doesn't start from nowhere. It actually takes in our bodies, actually, I'm just going to ask a patient advocate, how long do you think cancer takes to grow in your body?
A long time. It depends on the cancer.
Oh gosh, that's far too informed. Okay, I need somebody else. Any guesses?
15 years.
Oh, that is very well informed. So actually a lot of people think that cancer only takes a couple of years, but the truth is it takes a lot longer. So in the cases, for example, of colorectal cancer, which isn't up here, but it can take up to 30 years to develop in our bodies. And we know from the work that's been done in the past trying to prevent cervical cancer that you can actually see these early changes, these CIN changes, they're actually pre-cancer changes, and they start to occur about 5 to 10 years before you get invasive cervical cancer. Ovarian cancer, the ovaries are well hidden. There's nothing for you to physically see with your eyes, but we now know that it starts in the fallopian tubes. Ahmed's up there in the audience. He knows this more than anyone. It takes 4 to 6 years, or perhaps even longer. Would you agree, Ahmed? And lung cancer, 10 to 30 years. So actually, when we treat cancer, we're actually treating the end of a disease process that has already been going on in our bodies for sometimes decades. So why are we focusing on above the waterline end stage of a process where we're missing the main part of the action? We're missing it in this pre-invasive stage, which, as I said, is visible in some contexts. You can see it on the cervix, but in most cases, it's fairly invisible to the naked eye. It's invisible on scans. There are currently no blood tests for detecting it. But why are we not thinking about going into moving our research and focusing instead, sorry, this makes you dizzy, on pre-cancer? And now this is a phenomenon, pre-cancer, that doctors have known about for a long time. We know that there's such a thing as pre-diabetes, don't we? We also know that there's the equivalent in cancer. But oncologists like me, and even latter-day oncologists, know that our focus has always been on cancer. We are cancer doctors. We don't work in pre-cancer. It's kind of outside our area. But then who does? Who does work in pre-cancer? So I think we need to own it and we need to understand it. And there is work that's been going on in the States looking at really trying to understand what precancer actually is. We know that it's very different. So if you think about an invasive cancer, it's invasive because it invades through the basement membrane. You can see that it invades down through into the tissue below and gets into the bloodstream and it spreads to other places. It's metastatic. But one of the things we know about precancer is it has not yet got to the point of invading into the tissues or spreading to other places. And it's also very interesting that precancer is not a one-directional thing that doesn't necessarily always go to become cancer. We know that actually precancer can revert to normal. Our bodies are constantly fighting precancers. Our immune cells are doing exactly that. So first of all, we need to shift our focus. So we need to stop solely working, and I'm not saying we're not going to carry on working in treating invasive cancer, but we need to actually shift as well and start including our focus, our research focus on pre-cancer. At this early stage, it's still reversible at this point, but it's probably, we're looking at sort of 5-10 years in as part of it, towards developing invasive disease. So is this a better time to intervene? I've spent my career looking after people with very advanced stage disease, testing drugs in people whose prognosis is less than 6 months. And I can tell you it's heartbreaking because by then that their cancer has learned loads of mechanisms to evade treatment. So it becomes very, very resistant and it becomes very plastic. It's able to move. As you throw a treatment at it, the cancer learns a resistance mechanism. So can we go earlier? Is this a better time to intervene? Are we dealing with a different type of disease process at this point? And one of the things that is really interesting to us is that it's been known for a long time that there are things called HLA molecules on the surface of normal cells, which are a little bit like semaphore flags. And they present self-antigens to the immune system. So basically what happens is the normal cells, as in green, will always talk to our immune cells and say, I'm fine, I'm fine. The immune cells look at that, they go, great. But if there's an abnormality in the cell, if they're virally infected or if they're carrying a genetic mutation, suddenly they recognize that something's not right and they present these altered neoantigens, these altered antigens, proteins to the immune system. And they say, I'm not right, something's not right here. This is what is going on in our bodies right now and all the time. And then T cells come in and they recognize and bind to those abnormal regions and they destroy the cell. So that is our body's way of mopping up abnormal cells. What we've discovered is that you can actually design vaccines against these neoantigens. You can actually stimulate the immune system to better recognize abnormal semaphores as they appear. And this has been done extensively actually already in cancer. So we know that cancer cells also produce many of these altered neoantigens. And so vaccines that have been developed recently have been designed to treat established cancer. And this is great. But the problem is that the cancer cells have already developed mechanisms to hide, immune evasion mechanisms to hide. So that actually the immune cells are not very good at binding and attacking and destroying cancer cells at this late stage. So cancer vaccines are okay, but you need to give them in combination with other immunotherapies to stimulate their response. And even then, they're only shifting the dial marginally. So you have to give them, for example, and you have to give them alongside immunotherapies. But what about precancer? What about this stage I was talking about, which is this stage before the cells become invasive? This early stage, this transitional state that can be in our bodies for 5 or 10 years. What about precancers? Well, interestingly, we find that they are also presenting these abnormal semaphore flags to the immune system. But the cells, but at this stage, they have not learned these escape mechanisms. So your immune system is actually very good at recognizing them and destroying them. And that's how our body is constantly fighting precancer, because it's recognizing these changes on the cell surface and the T cells are in there and they're destroying those cells. And here you have cell death, so you see a return to normal. But as you get older, or if you have cancer predisposition syndromes, your immune cells are kind of overwhelmed. And partly it's because you have a sustained burden, many precancers in your body. And as you get older, also your immune cells are less good at recognizing those abnormal semaphore flags. And they're less good at interacting and destroying those cells. So the policy behind our vaccines is, could we give vaccines to people at this stage when they're likely to be harboring a precancer and use the vaccines to activate those slightly lazy T cells and see if we can get the body to properly eradicate precancers at that point. So this is what we're talking about when we're talking about, our prevention vaccines. They're designed to recognize or just teach the body to better recognize these antigen flags in a way that they should already be doing. So it's not trying to give something completely different to your body. It's really using your own immune system to better defeat precancers. And that's the strategy. And actually, we've got the immunopeptidomics team on here. And one of the skills that we have in OxCODE has been able to get hold of these precancers. Actually, I was saying how difficult they are to find, but we've got really good links to the clinic now. And we're able to get hold of precancer tissue. And using a very clever method that has been developed in Oxford as well as in other places, we're able to actually interrogate what the semaphore flag actually contains. And then we design a vaccine that exactly replicates that message to teach your immune system every time you see that abnormal signal, it's going to go and send some T cells and destroy the abnormal cells. So the first one out of the gate is LungVax, which is a vaccine that we've been working with UCL and the Crick Institute in London. And really this work came from Professor Sam James and also the Tracer X team at UCL. And what they discovered is that smokers and people who are at risk of lung cancer do actually have these changes, I talked about before, these abnormal semaphore flags. They can actually pick them up in the airways of smokers way before they get cancer. And then if you get a group of smokers in the room together, they will have the same semaphore flags as each other. So you could actually design one vaccine to work against all of those people. You wouldn't have to personalize it at all. And so what we have done is work with them to design the LungVax vaccine. And we're working also with a team in Oxford who, like, for example, Dame Sarah Gilbert, who developed the ChAdOx vaccine, which was used during the pandemic, the Oxford AstraZeneca vaccine. So that's a chimpanzee adenoviral vector. And instead of being anti-spike, we've taken that out and we've made it anti-neoantigen. And the idea is very similar. In fact, I just modified the ChAdOx picture here, that you vaccinate somebody and then essentially the neoantigens get expressed in their dendritic cells, in their lymph nodes, and stimulates the T cells, who then start hunting for any precancers they can find. And what we expect is to get a lasting effect by doing that. And so we have designed a study, which is a precision prevention study, which is a sort of dedicated study run in a high-risk group. And to start with, we're putting 40 patients in, 20 who have had a small lung cancer resected already within the last 3 months. And those patients we know are at quite a high risk of either a new cancer appearing somewhere or a recurrent lung cancer. And then we're also putting in another 20 who are at risk of lung cancer because they're heavy smokers or because they have other risk factors. And we're going to be testing the vaccine, and we're calling it ChAdOx2 LungVax, in that group, those two groups, looking first of all at its safety. Is it safe? What are the side effects? Well, we know that ChAdOx itself was given to over 2 billion people during the pandemic. So we're quite happy with the vector. But what about LungVax? What about that string of those neoepitopes we put in there? How safe will they be? And importantly, do they stimulate immune response? We hope they do, of course, but do we find that these are quite elderly patients who don't have a very good T cell response? So for us, there's lots of interesting biology that's going to come out of that very early on. And depending on the outcome of that, we're going to go into a large phase 2 study where we'll be looking at whether or not it actually does prevent cancer in that group, in those groups. And excitingly, we've now got the vaccine ready to go. So it's in vials and ready, which is a kind of moment that kind of, I don't know, it's really, when we first sent this picture from the biomanufacturing facility in Oxford, we were all in tears because it was stunning to actually see this concept in reality. And we heard last week that CRUK, some of whom are in the audience, have funded the project. So the study will be starting next year. So one question that we also want to ask is, should this vaccine be DNA-based or should it be mRNA-based? So ChAdOx, the one I'm talking about, is essentially a DNA-based vaccine. And the design of it is such that we have put those neoepitopes I talked about, those beads I showed you, those are going to be in sort of format of DNA enclosed within this chimpanzee adenoviral vector envelope, which is you can see the capsid thing there, the sort of apricot-colored dots around it. They then enter the cell, they release the DNA, which gets incorporated into the host DNA, into our DNA. That makes messenger RNA, which then gets turned into protein, which is then expressed on the surface of the dendritic cell, and that's what stimulates the T cells. This is all very technical. You don't really need to know this. But I'm telling you because the mRNA vaccine is another choice. And you hear about mRNA vaccines, and essentially the endpoint is the same. They both converge in the same area of basically enabling the dendritic cells to express the neoantigens on the cell surface. So this then stimulates the T cells. But the mRNA is a slightly shorter course. It doesn't go through the host DNA. It goes directly into the cytoplasm of the cell and makes the messenger RNA quickly gets converted into protein. And there are different advocates. If you speak to half the people in the room, like Ellie over there, who will I hope will ask the questions later, they are strong believers that ChAdOx may be better or have a stronger response. And then others would say, well, the mRNA is better. So there's two different schools of thought about that. And we thought it'd be interesting to actually do that comparison in this study. So we're working with a commercial company who's at the moment generating the mRNA equivalent. And so we will have a cohort who will be probably the adjuvant cohort, probably the patients who've recently had surgery. And we'll probably be we're going to be using an identical LungVac strip, but this time it'll be mRNA, not within that chimpanzee adenoviral vector. So this is kind of an extension to the LungVax project, which may extend it by an additional year, but it's a really important comparison for us to make at this early point. Which is the safest? Which has the strongest immune response? Which has the most lasting immune response? So they're important questions. And there have been very many people involved in this project, including, I'm delighted to say, we've always had patients in the public from very early helping us kind of think and conceptualize the study and what is acceptable and what isn't. And we've been extremely lucky to work with some very prominent people at UCL and the Crick Institute, including Professor Charlie Swanton, who's quite famous for his work in lung cancer, and not to mention the fantastic teams in Oxford. So obviously we're a little bit ambitious. We're not just going to stop at lung. We also want to do a similar strategy, apply a similar strategy, and develop vaccines for people at risk of colorectal and endometrial cancer. And these people have Lynch syndrome. And we'll hear a little bit about that later on. So we're developing, using a similar strategy, a vaccine that we can give to these people whose risk of colorectal cancer is around about 70 to 80% in their lifetime. So really, that is a sword of Damocles. And if this vaccine was effective, could these people be protected from needing all the chemotherapy and all the horrible surgery, including sometimes having their colons removed? Can they be prevented from that in their lifetime? Similarly, we're also looking at a vaccine to protect people who carry BRCA mutations. It's another hereditary cancer predisposition syndrome that means that people have a very heightened risk of both breast and ovarian cancer. And at the moment, the options are basically surgery or no surgery. Surgery or, you just live with the risk. And surgery means having double mastectomy age 30 and then both your ovaries and fallopian tubes taken out at 40. That's quite a significant operation to have, you're basically neutering people because of their risk of cancer. Could we give them a vaccine to protect them instead? And then there's other work, I've called this cheekily colorectal vaccine. It's a long way from being a vaccine, but we're doing some discovery work to see whether the non-Lynch associated colorectal cancer, the more common variety, can also be protected with a vaccine. Ultimately, what I would really like to do is to put all these vaccines together in one single vaccine. I'm calling that all-vax, because I have no imagination whatsoever. It's getting a bit desperate by this point. And this idea that you could protect everybody, that our children might one day go and have a vaccine aged, I don't know what, we need to talk about that, age 30, to protect them, so that cancer does not cause that destruction and damage that it has to so many of us and to so many of our lives. So to conclude, cancer incidence is rising globally, and we urgently need new approaches. Pre-cancer sitting there under the waterline, it's hanging around for a decade. Why aren't we doing more? We need to get in there. We need to intercept and prevent cancer. We discovered that these pre-cancers are fascinating because they present signals to the immune system that we can grab and we can turn into a vaccine. And then these vaccines can be targeted to destroy precancers before they even start and prevent cancer from even beginning. The first of these is LungVax. We're designing it to prevent lung cancer in people at risk. We're looking at mRNA versus using a DNA approach because we'll never get the chance to do that again. And we're also opening the study against Lynch-associated colorectal and endometrial cancer, and that's planned to also open next year. If effective, could we have an all-vax vaccine? Could we make cancer a disease of the past and not of the future? Thank you.